Well, hey guys, here's our next section of notes. This is section 3 of our notes. This is going to be the muscle filaments. We're going to primarily be talking about muscle filaments, but we do have a little bit of a discussion here at the beginning about exercise and what happens to our muscles during exercise. So let's get started. Hey, remember, this goes on the input side of your notebook on the right-hand side, and the activity that we do in class that complements this set of notes will go on the left-hand side and the output side. All right, so get something to write with and let's get going here. Your essential question for today is, why are I bands lighter as compared to the A bands in your muscle fibers? All right, as we get through these notes, you will be able to answer this question, so make sure you do that before you finish up. All right, so as I mentioned, we're going to finish up talking a little bit about exercise, where last time we were talking about hypertrophy and the different stages or the different forms of hypertrophy that your muscles can go through. All right, so exercise-induced muscle damage. Any of you ever have any issues that have occurred because of your athletics or things like that? It can really, really hurt when this happens. All right, so intense exercise can cause some muscle damage, which I'm sure you guys can all attest to that, especially if you're not getting the appropriate amount of nutrients. Electron micrographs reveal that torn sarcolemmas are damaged myofibrils and disrupted Z-discs. Okay, we can see over here in this picture, we've got normal muscle tissue is more of a lighter red color. All of the muscle fibers appear to be where they're normal, okay? They're where they're supposed to be. Now, strained muscle tissue, we see the muscle fibers are enlarged and they're redder, which means they're getting more blood flow to them because they're being strained, okay? Some of them may have ruptured a little bit or been disrupted where the Z-discs are, and we're going to talk about what this means, the Z-discs. Like I said, increased blood levels of the myoglobin and creatine phosphate are found only inside the muscle cells. Now, muscle soreness can happen like immediately, but oftentimes we notice that there's a delayed onset of muscle soreness. 12 to 48 hours after strenuous exercise, which leads to stiffness, tenderness, and swelling due to the microscopic cell damage that we see. Earlier this week, or last week actually, I told you it had to do with a buildup of lactic acid. That is true, but what happens is when we have this buildup of lactic acid, we have some other issues that happen. So here you go. As you weight train, now this is like intense exercise, strenuous exercise, you cause micro trauma or damage to the small fibers of your muscles, which are composed of proteins. Okay, so what's happening there is you're beating yourself up from the inside out. Okay. It's the repair to these microtrauma, usually by satellite cells. Remember what those are? Those satellite cells, they come in and do some repair that result in muscle growth in addition to added proteins, okay? So the harder or longer that you train, the more damage you do, which means you need to eat possibly twice as much protein as a sedentary or normal individual. Okay, so if you are weight training, you're probably feeling that soreness in your muscles tearing and ripping because of this micro trauma. Make sure you're replenishing those proteins with twice as much protein intake in your diet. Okay, so that could be in the form of shakes, an extra steak, or some other source of protein, okay? Then as your muscles adapt to your exercise, soreness does tend to decrease because the adaptation occurs and you're not causing that micro trauma like you were at the beginning. All right, now onto the filaments of the sarcomere and how all of the filaments come together, all right? Thick and thin filaments overlap each other in the pattern that creates the striations. So those stripes that we see in skeletal muscle and cardiac muscle are the result of the thick and the thin filaments overlapping each other. Okay? The lighter color bands that we see are called I bands, and the darker stripes are the A bands. All right? Now the reason why the I band region is lighter in color or lighter in appearance is because it contains only thin filaments, okay? Only thin filaments. And they're arranged in compartments called sarcomeres, which are separated by Z discs, all right? In the overlap region, 6 thin filaments surround each thick filament, okay? So let's put that into perspective. There are 6 thin filaments for every 1 thick filament. And these things are separated by Z discs, and from Z disc to Z disc, we see the sarcomere itself. Okay, so there's a new vocabulary term we haven't really defined yet. So a sarcomere contains these things going from Z disc to Z disc. Okay? All right, now to break it down a little bit more, the thick and thin myofilaments. All right, same picture we saw before. We've got the Z disc over here, the Z disc over here. Everything from Z disc to Z disc is the sarcomere. Now, the sarcomere is split in half by the M line, okay? All right, so now if we were to zoom in on this M line where the thin and thick filaments are overlapping each other, here's what we see. We have what's called the H zone. Then we've got zone overlap here and here. The A band is where we see the thick filaments, okay? And the I band is where we see just the thin filaments, all right? And the Z disc lies in the middle of the I band, really. And we see the M line rides in the middle of the A band, okay? Now, if you recall from our vocabulary, we talked about the thick filaments and the thin filaments. Myosin is our thick filament. Actin is our thin filament. Okay, just to bring that back into your mind. We do have supporting proteins that are also involved in this, and that's why whenever you are exercising intensely, you need to replenish that protein. It's because we've got a lot of protein interaction here. Supporting proteins occur on the M line, the titin filament right here, which is kind of an anchor filament that kind of rides between everything and it coils up between muscle or between these other filaments. And the Z disc, they help anchor the thick and thin filaments in place. Okay? All right, here's kind of a microscopic image in black and white of the exact same thing that we were looking at. The overlap of thick and thin filaments within a myofibril. All right, the dark bands are the A bands. So this is the A band, and this is where our thick filaments are. Then the light bands, these are the I bands, right in here, which are split down the middle by the Z disc. The H zone, recall what that was. That's kind of the zone between overlaps of the actin and myosin filaments. Okay? And this is an electron micrograph. So it's not exactly a microscope, it's an electron microscope image. This is zoomed in on 2 sarcomeres. So we see one sarcomere goes from Z disc to Z disc, so we've got 1, 2 in this particular micrograph. All right, hey, this is our last slide for today. Awesome. Okay, so the proteins in muscle. You're going to be doing a protein tree as part of your assignment that goes with these notes, and you're going to break it down into some more information, okay? So this will get you started. Your proteins in muscle, your myofibrils are built of 3 kinds of protein. The first kind here are the contractile proteins. All right, so you got contractile protein. Whoopsies, let me go back. I don't know what just happened there. Contractile proteins. I want to put this in black real quick. Okay, here we go. Your contractile proteins, which are the myosin and actin, which give us those thick and thin filaments. Then we have our regulatory proteins. So this regulatory, remember that whole big H word we learned at the beginning of the year, helps with regulation. I can't keep this straight. All right, so regulatory proteins. This would be to maintain homeostasis, right? Which turn contraction on and off, which are your troponin and your tropomyosin. All right. Then we have our structural proteins, which are there to support. They provide proper alignment, elasticity, and extensibility. Okay? And this would be your titin, your myomycin, your nebulin, and your disotrophin. All right? So these are the proteins that you're going to start with on your protein tree. Take a look at my notebook. I've set it up so that you can get busy with it. And there will be some detailed instructions for you that I leave so that you can check it out and get busy with this inquiry activity. Hey, this concludes your notes for today. Make sure you go back and answer the essential question about I-bands and get that into your notes. And don't forget, we're doing this in Cornell note fashion. Include your summary and the questions in the margin. All right, thanks guys. Have a great day.